In this section, I'm going to provide an introduction to mycology. So there's approximately 3 to 4 million species of fungi, and they can be divided into two main groups: macroscopic, which means that you can see them with the naked eye. Those are mushrooms, puffballs, gill fungi. And then microscopic fungi are the molds and the yeasts. If we look at them, we have the yeast cell, hyphy, and some are dimorphic. So yeast cell is round to oval shape, and they use asexual reproduction. Yeast are single-celled organisms. Hyphy are long thread-like cells found in filamentous fungi or molds. If you ever look at a piece of moldy bread or moldy anything, it usually looks fuzzy. All those fuzzy strands that you see, those are hyphy. And then pseudo-hyphy are chains of yeast cells. So sometimes yeast cells can form chains. It doesn't mean that they're multicellular, they're just existing in chains, and then they can break free from those chains. Dimorphic means that some fungi can take either form. So for certain species, especially medically relevant species of fungi, if you incubate them at body temperature, they're going to grow as the yeast form. So they're going to be single-celled and look like the yeast form. If you grow them at room temperature, they're going to grow in the mold form. So they're going to look fuzzy. And that same organism is the same species. So it's kind of crazy if you think about it. Just the incubation temperature can change how those organisms grow. This is a picture of a typical yeast cell. So yeast cell is going to have all those structures that we just talked about in eukaryotic cell structure. And yeasts will replicate by budding. And you can see a bud forming off of a yeast cell. So this yeast organism is a large organism. And then instead of mitosis, where it splits in half, where there's two equally sized daughter cells produced in mitosis, you have this little tiny bud that buds off. It breaks free, and then it'll grow up to be a full yeast cell. Yeast are different from bacteria. When you look at them under the microscope, a yeast cell is a lot larger than a bacterial cell. It's usually oval shaped, and if you look closely, sometimes you will be able to see the buds. And here's a picture of an electron micrograph picture of some budding yeast, and then a cartoon drawing of yeast with buds. Fungal nutrition. So how do they eat and what do they eat? Well, fungi are heterotrophic. Again, break down this term, heterotrophic. Troph is talking about eating. Think about a food trough. Hetero means something else or different. So it eats something else. It eats a different organism. It's not able to be autotrophic. Auto means self. That would mean it is able to make its own food source. So heterotrophic, it has to eat something else in order to survive. So it acquires nutrients from a wide variety of substrates. Saprobes. A saprobe is an organism that's going to obtain substrates from dead plants and animals. Basically what it does is it sticks to the surface of the dead plant or animal or other type of organism. It will spray out digestive enzymes and then absorb the nutrients. If you think about if you've ever had a fungal infection, if you've had athlete's foot or maybe had ringworm or even dandruff cupitis is and also known as cradle cap in infants. Those are all types of fungal infections. If you've ever had an infection like that, you notice that the skin becomes inflamed and red. The fungal organism is on the outside and it's spraying digestive enzymes on the surface of your body. And then it's absorbing those nutrients. So that's why you see it looks really inflamed, because it's literally dissolving you and digesting you from the outside in. Parasites. So parasites live on the bodies of living animals or plants. Some fungi are parasites. And when I just talked about how if you've ever had a fungal infection or know of anyone that has, that would be a parasitic infection. Saprobes, that's a natural thing they should do to decompose dead organisms. Parasites, that's not so great. So this is a picture of molds growing on dead raspberries. So you can see all of these little strands here, those are hyphae. In order to properly be able to visualize them, you'd have to use a microscope to see them. And then here is athlete's foot, or a fungal infection of the foot. Most microscopic fungi grow in loose associations or colonies. So we have mycelium. Mycelium is the woven intertwining mass of hyphae that makes up the body or colony of the mold. So usually when you look at a mold growing, there's this central, really dense component of the mold, and that's the mycelium. The mycelium is that central, dense component. The hyphae will then intertwine inside of there and then extend out to form those hair-like structures that stick out. Septa, those are cross walls dividing hyphae into segments. Fungi, especially when we talk about diagnosing fungi in a medical setting, they will be classified as septate or non-septate. Septate have those cross walls in their hyphae, non-septate do not. So sometimes a hyphal strand will just come out and it has no dividing walls. That is aseptate. Sometimes the hyphae branches out and then it has dividing walls in here and they form individualized cells in here, and that would be septate. So septa are talking about all of these little walls that you would find in the hyphae. And hyphae, again, these are the strands of fuzz that you see growing on the surface of the fungi. This is important medically. Like I said, in order to diagnose some fungal infections, we actually look at the hyphae under the microscope and determine whether it's aseptate or septate, and that will help give us some distinguishing features to diagnose. And then spores. Spores are another way that we diagnose fungi. The shape and location of the spores, whether they're coming out of certain types of phyllidase or other types of spore-like structures, that helps us identify fungi. So spores are fungal reproductive bodies. Sometimes I refer to spores as the seeds of the fungus. So you can plant a seed in the ground and it'll grow into a plant. You can plant a spore into the ground and it will grow into a fungal organism. So this is kind of a diagram of vegetative versus reproductive hyphae. Vegetative, whenever you hear vegetative, it's just the living, breathing, moving, active form of the organism. So this is your everyday hyphae. And reproductive means that it's trying to make babies. So it's going to be releasing spores. So if it gets into its reproductive form, it's going to pop up this bud, and then it's going to release spores from this bud. Mushrooms. If you think about a mushroom is a fruiting body of a mold organism. The mold is extensive and it grows underground. And when it wants to reproduce and release spores, that mushroom will pop up. From the bottom of the mushroom cap come all of those spores out in the environment. So fungi have many complex and successful reproductive strategies. So simple outward growth of existing hyphae, sometimes they'll just branch off and that's your fruiting body. Fragmentation, so separated piece of mycelium can generate into a whole new colony. So you just pick off one piece of the mold organism and then put it somewhere else and it will grow into a whole mold organism. The primary reproductive mode, though, is spore formation. So these fungi will produce spores. Fungal spores should not be confused with the more resistant, non-reproductive bacterial endospores. So we'll talk about bacterial endospores a little bit later, but fungal spores are not very resistant to environmental stressors. You don't need to know all of the different types of spores. Now, if this were a medical mycology course, then I'd expect that you be able to know all the different types of spores and be able to identify fungi based on their spores. What you need to know is that there's two main types of spores, asexual and sexual. Asexual, A is the absence of. Sexual is talking about sexual reproduction in which two gametes come together to form a new organism. So asexual, it's just when one organism produces an exact copy of itself. Asexual spore formation is very common with fungi. So this is the predominant mode that they reproduce. One mold organism will grow a reproductive structure and it'll spray all of its spores out of that reproductive structure, and those spores are identical copies to the parent organism. They will go off and make identical copies of that organism. There are sexual spores as well, and sexual spores are pretty rare. They do include some relevant or fungal organisms, so they are important, and they're going to increase diversity. A sexual spore has to fuse with another sexual spore in order to produce a new organism. That fusion causes combination of genetic material, which again increases variations. So slight variations in the form and function are potentially advantageous to the adaptation and survival of the species. If all we did was make exact copies of themselves, and then we had a sudden change in the environment, if we didn't have diversity, every organism may not be suitable to adapt to that new environment. So having diversity increases the likelihood that some organisms are going to survive. Fungal identification and cultivation. So how do we identify medical specimens? We isolate it on special media. This special media is usually sabro-dextrose agar, and we put antibiotics in it, and those antibiotics prevent the growth of bacteria. If we have a nutrient medium, bacteria also want to grow there, but we don't care about the bacteria. So if we put antibiotics in there, it'll kill the bacteria and only allow the fungi to grow. Fungi take 4 to 6 weeks to grow at room temperature. Which is 25 degrees Celsius. That's a really long time. So when we're culturing these microorganisms, we will culture them, put them in an incubator for 4 to 6 weeks at room temperature. And then once they've grown, we can observe them microscopically and macroscopically. So macroscopically, we look at how fuzzy the colony looks. We look at the color of the top of the colony, the color of the bottom of the colony, and we can investigate how that looks. So microscopically, we will look under the microscope, and there's different types of staining procedures that we can use that are safe that will allow us to look at this organism under the microscope safely. One of the major stains, and I'll write it down here because it will come up during the lab portion of the course, is the lactophenol cotton blue. Stain. Lactophenol cotton blue will kill mold organisms or kill fungal organisms. So it's safe to use it that way. Fungal spores are highly dangerous. So if we're going to culture microorganisms, we have to culture it in a negative pressure room, and we have to take all of these different precautions. In the next segment of lectures, I'm going to talk about fungal identification in a medical setting and talk about some medically important fungal specimens, and I will get more into the dangers of working with fungi, how you culture them, how you diagnose infections, and so forth. So lactophenol cotton blue will kill those spores. It's the safest way to work with fungi and investigate them microscopically. Asexual spore-forming structures and spores are used to identify genus and species, along with the hyphal type, colony texture, pigmentation, physiological characteristics, and genetic makeup. And this is a picture of representative fungi. So here you can see hyphy, and then you can see one of the fruiting bodies, and all of these little dots are spores that are released from the fruiting bodies. Here you can see another type of fungus. You can see a hyphy leading up to a fruiting body, and the spores are emerging from the top of this fruiting body. Some medical conditions caused by fungi. So most human fungal infection occurs through accidental contact with soil, water, or dust, especially gardeners that are immunocompromised, that go dig in soil in warmer areas, such as down south and such. They have a higher risk of being exposed. Pathogenic fungi, there's community-acquired infections, which are caused by environmental pathogens. Hospital-associated infections caused by fungal pathogens in clinical settings. Most commonly, it's going to be a fungal yeast infection. And then there's opportunistic infections, which are caused by pathogens infecting already weakened individuals. So, opportunistic infection is a pretty important term that I'm going to spend a little bit of time defining right now. Opportunistic. If you had the opportunity to do something, that means that all of a sudden you're able to do it, that barriers have moved out of your way and now you're able to do it. Let's say now you have the opportunity to go to school. Maybe before you were working too much, or you had family obligations and that type of stuff, but something changed and now you have the opportunity to go back to school. An opportunistic pathogen is an organism that can be in a human or in an animal. It can be present in a host organism, but it's not causing infection yet until it has the opportunity to. So maybe that host, maybe that person, all of a sudden has an autoimmune disease or has diabetes, or maybe they have a weakened immune system. They had cancer and they went through chemotherapy. Well, now this pathogen that has been hiding out inside the body has the opportunity to overgrow. It will grow and grow and grow and cause a major infection because it had that opportunity. We refer to those as opportunistic pathogens. In a normal, healthy individual, they don't cause infection because they're kept in check by your healthy immune system, by the secretions inside of the body that keep that organism at bay, and by other microorganisms, the good, beneficial microorganisms that are there to help protect you. But they suddenly get the opportunity because one of those protections goes away. So opportunistic pathogens, we're going to talk about quite a bit in this course. Other medical conditions caused by fungi, allergies. Allergies are extremely common. There's a lot of people with mold allergies or spore allergies, and that's very common. And then there's neurological conditions due to toxin production. But there's some benefits to having fungi around. So they play an essential role in decomposing organic matter and returning essential minerals to the soil. They form stable associations with plant roots that increase water and nutrient absorption. So they get food from the plant, but they provide the plant with some extra water. Production of antibiotics, alcohol, organic acids, and vitamins. So they're used to produce a lot of different things. We talked about antibiotics and why fungi produce antibiotics before. Food flavoring, and alcohol in beer and wine, gas that causes bread to rise.